Plant diseases can be identified in several ways. Often, different plants have specific diseases they're susceptible to, and the visible symptoms can be looked up in a gardening book or online. What other methods can we use to identify the disease that may be affecting a plant? Identification can also be done by scientists in laboratories. People managing infected plants will take them or a specimen to a specialist plant clinic containing a laboratory. So what happens in the laboratory? Within these laboratories, lots of different tests can be carried out on the plant. These laboratories will have scientists that are experts in testing plants and identifying plant diseases. Laboratories are also likely to have access to specialist equipment and data to identify the disease. You don't need to explain how different pathogens are identified in laboratories, just that this is a method that can be used. Another way of identifying a pathogen in a plant is by using testing kits that contain monoclonal antibodies. Specific pathogens can be identified using testing kits that contain monoclonal antibodies. The antibodies will attach to certain pathogens and mark them. When the pathogen has been marked, this makes them visible to the person doing the test, commonly appearing through a mark appearing on the kit, which is a positive result. For the exam, you don't need to describe how these kits work, just know that they utilise monoclonal antibodies.
Thanks for watching. If you want to take your GCSE revision to the next level, head over to launchpadlearning.com and check out our smart learning platform that's been designed to get you top results in your exams. We cover your whole specification and make revision fun with interactive quizzes, easy-to-follow videos, and more. You'll be kept motivated by your own AI tutor who's here to support you every step of the way. To check it out for yourself, click here. Or click here to keep watching a selection of the videos from our full GCSE biology course. See you there!